Hello, everyone, and welcome to today's webinar, Debunking Antibody Titration Myths. I am Julie Abing of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by SciTech Biosciences. To learn more, visit scitechbio.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd now like to welcome our speaker, Deanna Bonilla, Global Scientific Director of SciTech Biosciences. Deanna, you may now begin your presentation.
Well, hello, everyone. Thank you so much for joining us today. This talk is about a simple but relevant topic, antibody titration. We all know how critical antibodies are for flow cytometry, and part of that depends on titration. So the goal for this presentation is to show you how titration is key and also to address some questions and misconceptions around the topic. So if we start with kind of how the presentation is organized, I divided it into two topics. The first section is going to be an introduction on titration best practices. And the second part is going to be some comparisons we did for over 100 titrations across different variables, different conjugates, specificities. So we will go there later. Antibodies are so useful for many technologies, right? Western blot, flow cytometry, immunohistochemistry, ELISA. And in terms of the structure, it's the same. Immunoglobulins have two heavy chains. You see them in yellow, two light chains on the sides. Also, as part of the structure, immunoglobulins have variable regions. Those variable regions are important for binding to specific antigens or epitopes within the antigens, and the constant region more for functionality. There are multiple or 5 subclasses or isotypes of immunoglobulins. They are different in their functions, but also, as you see for IgM and IgA, for their different number of antigen binding sites. Let's first review some important concepts before we dive into titration. This is the binding of an antibody and an antigen. That's what we call an immune complex. So for the immune complex, important things are the specificity. We want an antibody to bind specifically the unique epitope in the antigen. In terms of affinity, that's the strength of that binding. Avidity, also the overall strength, so depends on affinity, but also on anything else that can help stabilize the complex. Things like how many antigen binding sites are present in the antibody, how many binding sites in the antigen, anything that could help the molecules interact, things like having your antigen immobilized, and other factors, more technical ones like temperature, time of incubation, and staining conditions. When we talk about validation of antibody, it's kind of good title, right? Antibodies are not magical. It's not just getting the vial and start using it. It's all the validation that might be needed. And related to that, there are 3 publications in Nature the last decade that highlight the issues with using antibodies that are part of the reproducibility crisis that we see in science. But the key point and message from those publications is that you want to test your antibody in your cells of interest under the staining conditions that you plan to use in your samples, and in order to verify that it's performing well. So back in 2016, a proposal was signed by more than 100 investigators to have guidelines on antibody use and validation of these reagents. So in response to that, there's so many portals now or websites with information on how to use antibodies, but also validation data, which is so critical. So that's the place where you could start identifying clones and antibodies to use. What are the issues with antibodies? And kind of summarizing what was described in those publications, main issues are 3, at least the ones reported by them. Cross-reactivity, your antibody binding not only to your protein of interest, but something else, specifically or non-specifically, through the antigen binding sites, the yellow portions, the orange portions, or through the FC portion in the constant region. variation across lots with different performance, or just picking the wrong antibody. We know, for example, for Western blot, you are going to be detecting linear epitopes versus for flow cytometry, more of the native form of the protein. But even if all of that is controlled, we have seen how in flow cytometry, there's differences in performance for antibodies, and that's related to other factors, right? What clone you're using, not only because that's what's giving you the specificity, but also because some clones tend to aggregate more, for example, the conjugates, the type of fluor, but also its stability, the cell type, that's a critical part, that's where the antigens are going to be expressed for then antibodies to bind. Staining protocol, because anything in the staining protocol could affect that binding capacity, things like temperature, incubation times, washes, all the buffers that could affect the binding, but also the levels of expression, and definitely titration, and that's why it's the center of this presentation. As you see on the right side, on the bottom, there is some publications that have addressed how critical titration is for high quality data. So what I've done is summarize in 4 points why you definitely want to go for titration or complete titration for every reagent. Higher resolution, you're maximizing your positive signal, minimizing the noise. Also by increasing reproducibility, because you have consistency in the protocol that you're using, and that including the concentration of antibody that is used. The specificity of your staining, because you reduce the chances to have cross binding or non-specific binding by optimizing the concentration. Also related more to technical things. Your signal is going to be on scale, less spillover spreading between fluorochromes since the signal is dimmer, and also less aggregates, antibody aggregates, and also the chances to have your target cells aggregating because of the presence of those immune complexes. And something more not related to data quality, but more to the efficiency of the assay, which is saving money. Most of the time, the optimal titer is less than what the recommended concentration. So if we move forward, one of the questions that we get is, when should I be titrating my reagents? So the answer for sure is if you have a new assay, and for every antibody and any fluorescent molecule, so viability dyes, functional dyes need to be titrated as well. Once you titrate or optimize your assay, you might need to retitrate if there is anything new lots, clones, conjugates, anything changing in the protocol, staining protocol that could affect binding capacity. And definitely if there is something suspicious about the data. Your refrigerator is not working, reagents were left in the bench, exposed to light, storage conditions, and all of that could be impacting your antibody performance. There is also a lot of publications that include titration data as part of their panel validation. And this is what our example, a panel optimized by Cytec 2 years ago. It's a 40-color panel. So here I'm just showing you the titration, how they look. We will go deep on how to analyze the data. But this is for the violet dyes. The same has been defined or explained in the manuscript for every single reagent. Just to show you one example, this is a titration for IgG. On the y-axis, you will see the intensity of the signal. On the x-axis, you will see the concentrations that were tested from 2 to 125 nanograms per test in this case. And we clearly see, using only 2, there is not a clear separation for the positives. But if you use 125, now we see better the separation. Important thing, if you want to use this as a resource, is that in the publication, now you will have a table that is listing every single fluorochrome, every single reagent with the specificity, the fluor, information about the vendor, and also what titer was used, once again, in nanograms per test, important to report as concentration. And important to consider not only the titration, great panel design, the staining protocol, all of that together will allow you to then generate data that is high quality. In other words, that the resolution, the separation for all the populations in the panel is optimal. So let's divide the titration process into 3 parts. What do you need to have ready before you even start staining samples or acquiring samples on your cytometry? So 4 recommendations, starting with sample quality. making sure you have enough positive events, that your sample for titration is a good representation of what you will have in your multicolor testing in your final experiment, that you pick carefully the clone based on validation data using all those portals, and that then based on sample characteristics or quality, you can decide whether you need to include a viability dye, block your Fc receptors, lyse your red blood cells, or include co-staining. That's highly useful if you're going to detect a low express marker in order to find clearly the positivity. So you could include a lineage marker, C3, CD8, to identify things like TCR gamma delta easier in the titration. In terms of assay design, a lot of considerations in the staining protocol. What's your stock concentration, antibody stock? What concentrations are you testing? What dilutions? The final staining volume should match what you will have in your multicolor sample when all the other antibodies will be added. Acquisition details, how many events you're recording, also based on the frequency of the population. What are your acquisition settings, ideally optimized for your cells of interest, and flow rate as well, that's something that could be predefined. If you don't know the stock concentration, you can reach out to the manufacturer and get information. If you're using a Cytec antibody or conjugate, you can go to our webpage and find the certificate of analysis. And with that, you will get a document. That document will look like this. And in green, highlighted the concentration. So that's where you start from. The other part of the preparation for your titration is understand how you're going to do data analysis. Which populations are going to be gated, how to define, for example, which descriptive statistics will be used, if you're calculating any metrics, how you will define the optimal titer, the fact that you can concatenate all the samples to have a better picture of the full titration, and also based on that, decide whether you need to test additional titrations. And finally, understand that titration, it's a dynamic process. When you, for steps 1, 2, and 3, when you are in a single color scenario, the background that you're getting will depend on the cells, autofluorescence, will depend on the instrument, what settings you use, and how much noise you're getting. And it's going to depend also on the antibody, how much non-specific binding you're getting, or even unwanted specific binding. So when you move into multicolor scenario, other things are going to play a role now that multiple antibodies are added together. Either spillover spreading between fluors or an issue with the binding per se, the accessibility of the target, and that's more towards steric hindrance. The point is that the final titer that you optimize in 1, 2, 3 can change. You might need to adjust the titer or even adjust the staining protocol based on the results, go for a final adjustment on the fluor that was selected for a particular antibody. There is also a lot of publications, you will see them listed on the right, that go through protocols step by step on how to do titration. So if you want a detailed protocol, I suggest you to go there. Here's more to describe the process and give you some tips. So we start with the stock. Let's say we are testing titrating a CD4 antibody, SK3 clone, conjugated to BYG781, one of our Cytec dyes. You're going to start doing serial dilutions. Most of them work in a range from 1 to 2 milligrams per test, down to 8. Things can change depending on the marker, the specificity. For example, for CD20, you might need to go higher. For CD62 ligand, you might be lower, around 100. So this might need to be adapted as a general recommendation. And in terms of tips, make sure that your cytometry is fully operational, that you're passing QC, that your settings are optimized. That will be, in our case, Cytec assay settings, that you find the stock concentration, again, through the manufacturer, but you keep consistency in what's going to be the final staining volume for all those titration tubes, and also consistency in the number of cells that are going to be used. Our suggestion is to go from 8 to 12, two-fold serial dilutions, and better results for us diluting and staining in plates. Try to report in nanograms per test or micrograms per ml concentration instead of just the dilution, mainly because stock concentrations can be different. In terms of staining, centrifuge the antibody vial to remove any antibody aggregates, taking the aliquot from the top. And important that you match the staining conditions that you will have in your multicolor sample, because anything in that staining condition or protocol that could affect binding of the antibody is going to impact the titer. So now let's see what you would do after you get your FCS files ready. First step is to evaluate sample quality. But I want to talk about the key concept that we will be describing from now on, and it's the optimal titer. The optimal titer is going to be the concentration of antibody that will give you the best resolution, separation between your positives and your negatives, or between your positive signal and the background. What I'm showing you here is that titration of CD4 BYG781. But what we are seeing is different from in terms of concentration, different from the numbers that I was telling you before, 0.5 to 64 nanograms per test. And we will see why that's the case. But going back to sample quality, what I wanted to show you is that for every file you want to check, that you can define your population of interest in that first scatter pseudocolor plot, that you remove from the analysis cell doublets, cell aggregates, that you use time to monitor the stability of the signal, that then you can define your negatives and positives, all the statistics needed, and that finally, if you have a spectral cytometry, you will be able to see for every detector in the system how much light is being captured and build those signatures. So you will know that the signature for your negative should have low background versus the signal for your positive, in this case, should match the expected emission from BYG781, as we see all that emission out of the yellow-green detectors. And a consideration back to that idea that I'm testing lower concentrations that I had suggested before is because when we did from 8 to 1,000, what we see is mainly saturation. The MFI, the median intensity of fluorescence for the positives, is at the same place. And we are not capturing the initial part of that titration curve. So for this case, we added additional dilutions, and that's something you might need to consider on the lower end or the higher end, depending on what you are seeing. But if we focus on the image or the concatenated image on the right, we are looking at what's key for titration. Negatives and positives. If we start with the positives, what we see is using 0.5, the first dilution on the left, 0.5 nanograms per test, what we are getting is separating, is the CD4, but it's not saturating. So this is separating concentration. You increase the concentration and you see how the MFIs start increasing, and we reach a point where there is saturation. No matter that if you are adding 1,000 nanograms per test, the MFI continues to be the same or close to the same. That's for the positives. So if we look at the negatives, if we look at the negative for 0.5 concentration, it's really tight, low spread in that population. But as soon as we increase antibody concentration, we see the spread of the negatives. So at the end, the optimal titer that gives me the best resolution will be the one that gives me saturation in the positive, high signal, and low spread, low MFI in the negative. Let's see now what metrics could help me evaluate the data and also define the optimal titer. You're going to have negatives and positives ideally for each titer. You're going to be using things like the median intensity MFI for the positive and the negative, and a way to understand how much the spread of the negative. So there's multiple metrics that have been used. Signal to noise ratio is one of them, dividing MFI of the positive by MFI of the negative. Stain index, you subtract those two and also divide by 2 times the spread of the negative in terms of robust standard deviation. The other one is separation index. Also accounts for the spread in the negative, but in a different way in terms of the mathematical calculation. It's going to give more weight to the right side of that negative histogram, because it's the one that is indicating actual separation from the positives. So it's going to calculate, or based on the calculation of the 84th percentile. And finally, something that we highly suggest, and is that together with a metric calculation for the separation, go with the percentage of positive cells that you maintain. With going lower in titer or higher, you maintain your ability to capture the positivity within your population. Back to that CD4 BYG781 titration. Now showing you the metrics plotted when we have the metric value versus the concentration. So what we are seeing here is just the median. Very difficult with the median to look at the negatives, the green line almost down to 0. But if I move to, for example, log scaling, I can see better how the negatives are also increasing in MFI. But again, metrics can give you just one value, can give you a better idea. Let's plot signal to noise ratio. We see how things are increasing because of the increase in the MFI, reaching a plateau, but then decreasing that value because the MFI of the negative is also increasing. For stain index, you can just plot the raw stain index or the log to see better the plateau, but same observations, right? An increase in that separation because an increase in intensity for the positives, and then the plateau and even a decrease because how the negatives are behaving because of the non-specific binding. Similar observations for separation index, also in log scale. So a lot of ways to plot this and to calculate from all of them, and we will see part of the comparisons is around metrics, but the observation here is that for all of those, the optimal concentration is 32 nanograms per test for this particular antibody. And also important to monitor the percentage of positive cells. For CD4 might be easy, right? It's easy to find what's positive even when you use 0.5 nanograms. So still separating the difference between separating concentrations and saturating ones once you reach the plateau. And we will see why that's critical in this slide. So if we look at the titration curve and the different portions, let's dive in, starting with the initial part where the antibody concentration is low, you are not getting sufficient staining, and you see the separation in the pseudocolor plot of the negatives and the positives. In this area, there's going to be a lot of influence by tiny changes in the protocol, including technical errors, pipetting errors, changes in the temperature, in the incubation time is going to be having more impact because of we haven't reached the saturation point yet. Then the next part of that curve is going to be the saturation, where you have the optimal staining. And that's where the optimal titer will lie. If we look at the pseudocolor plot, now we see how the positives are much better separated. So in this area, the impact of other variables like time or like incubation time will not be as high because you're still already at saturation. So tiny modifications, including technical technical errors, that's going to be not as impacting as if you are in a non-saturating concentration. And after that, when we see the decline, are the excess of antibody. You now are getting higher background. Separation is not improved. If we look at the pseudocolor plot, positives are almost at the same MFI. We were seeing that in the concatenated image as well. But now the negatives look different. There's a lot of spread. In 4, maybe not that critical. I know where the positives are still, but for molecules that are dimly expressed, that's going to have a high impact. So once again, the optimal titer is going to be right in that saturation phase. And the way you could calculate that, it's by finding a fit for that curve. Maybe by using a nonlinear regression, and the optimal titer tends to be like twofold. The concentration that gives you 90% of the intensity, highest intensity in terms of MFI. We wanted to analyze a lot of titrations, or more than 100. So we wanted to do this in an automated way and also less biased way. So we got help from FCS Express. They created a universal titration template for us that is now available in their website, where you just add the files, define what concentrations you're testing, define what populations of interest you have, and finally, it's going to do all the batch analysis and calculation for you, including things like those regression plots, right? The concentration versus the metric that we want to use for resolution, and then the calculation of what's the optimal titer based on the different metrics. That was highly useful because now we wanted to use that template in order to compare across things. The multiple things were compared, and I'll review in detail each of them. Main questions for all these comparisons was if the optimal titer is influenced by things that I do with my cells, by things that I do with my files, or by the type of file that I use. First, or the calculation. So the first question was, is the metric different or giving me different results in terms of antibody optimal concentration? The second question is if the settings used for acquisition play a role, if the data type, raw versus unmixed for our units, it has any impact, fact that we can extract autofluorescence, the type of sample preparation or staining protocol that you're using, and finally, if it matters that you use an Aurora versus a conventional instrument. So let's start with the metrics. And just to summarize, these are the metrics that we compare: signal to noise ratio, stain index, separation index, and always together with the percentage of positive cells. So let's go back to that, well, CD4, but this time a conjugate APC. We're seeing some difference from what I was showing you before with the BYG dye. In this case, APC is dimmer signal, so the positives are a lower intensity. All of the concentrations can separate the positives, but we know, we can see where, like around 16, that plateau, we are in that saturation phase. In the negatives, we don't see that much spread. It's given by the instrument per se, the detectors, and the wavelengths for these fluors. Anyway, the point is to compare metrics. So what you're seeing in this plot is all the curves for the different metrics. The dotted line on top is the percentage of positive. The secondary y-axis will guide in terms of the numbers. For all the color ones is the stain index, separation index, and signal to noise ratio. And again, for positivity expected, it's not changing. Even if you use 0.5, you can still find the CD4 positives. We will see with other specificities how that looks, especially for low express antigens. But the point for all the metrics is that when we define optimal titer, all of them gave me the same result in that linear regression evaluation. It was 32 nanograms per test, so similar performance for all the metrics. If I now show you, for example, same specificity, CD4, using clone SK3 in human samples, and now across fluors, APC, PE size 7, PE, fluors that have different brightness, like APC dimmer, PE size 7 brighter, and definitely PE our brighter fluor. What we are seeing here in every single plot, it's a titration, multiple concentrations, and once again, the progression of that titration curve. Very similar. Is the same antigen. This depends on antigens and antibodies. What the fluor is changing is how intense we are getting the intensity of fluorescence for the positives, and also the background, also related to the instrument. But in general, for all of them, since it's the same antibody, we are getting similar titers, similar needs in terms of antibody concentration to label at saturation certain marker like CD4. Important here now that we see how fluorochromes are going to be useful, depending on marker expression, to be able to resolve better populations. So definitely something dimly expressed PE would be the first solution, but a good option for APC or even something dimmer. But important that all that is taken into consideration, even though the same antibody concentration was selected, optimal concentration for this. Now showing you the plotting of that, all the metrics. So you see, same marker, the trend is very similar in where you're getting to that plateau, which is the optimal concentration as well, and how for CD4, percentage of positives is not that informative because you can still gain because of separating concentrations. So what if I have a marker that is not that highly expressed? Not a lot of cells are expressing it, like CD123. So what we are seeing is a titration from 16 to 1,000 nanograms per test, and now showing you the metrics. Percentage of positivity, so you see on the secondary y-axis is less than 1. So it's a different situation than CD4. But still, 16 nanograms can still give you a good estimation of that percentage, so not that helpful there. But if we look at the metrics, the trends, the shape of those titration curves is very similar. For all of them, what we got is that 250 for this particular conjugate is the optimal concentration in terms of nanograms per test. Let's now see a marker that is low expressed, like TCR gamma delta. So you won't be able to define the positivity that easily if you go on the lower end. And we see it, right? 16 nanograms, you don't clearly see the positives. You start getting better separation. 500 is the best because at 1,000 nanograms, you're getting an amplification of the background. So if I show you the metrics, we're seeing the same, very similar. All of them are giving me 500 as the highest. And we see a drop on that metric, both for stain index separation and even for the ratio. And that happens because of the background, right? We see it on the concatenated plot. That's why it's so critical to have everything, a metric, the concatenated plot to look at the actual data, and in this case, the percentage. We see how at 16 nanograms in that dotted line on the graph, it's almost half of your ability to detect the positives, it's reduced by half at low concentrations. And the highest is going to be around 500 and 1,000. So putting those two together when it's a dimly expressed antigen, it's our suggestion. Part of what we did was to understand the correlation between metrics. We were seeing that visually, but we wanted to get numbers. There was high correlation. This is for 4 different specificities, but once again, we tested a lot of titers and titration sets. There was good correlation. So based on that, based on the previous conclusions, and also based on the fact that stain index is easy to calculate, is widely used, we decided to move with only that one. We did the calculation for everything, but for the presentation purposes, I'm going to show you mainly stain index for the other comparisons. So metrics don't seem to play a big role, even though we were expecting, because the spread for some of them, or the negative, is taken into account. Now let's move to acquisition settings. What if I use optimal settings, or if I decrease the gains or increase them? What's the impact on the optimal titer? So here showing you CD4 data, 3 data sets. The one in the middle is optimal settings, acquisition settings, which are our Cytek assay settings. On the left, plus 50, when we increase the gains by 50%, and on the right, decreasing the gains by 50. Anything that goes out of that range, it's gonna, the conclusions might not apply, so just warning. What we are seeing visually is that, yeah, if I increase the gains, the positives are higher. If I decrease, the positives are dimmer compared to the one in the center, gold standard, or optimal settings. But also when we increase the gains, the negatives are also increasing in MFI and spread. Let's see now the metrics. So if I show you stain index first on the right, and I'm going to show in parallel stain index and positives, and you will see why. We part of saw it, but I'll explain why more. Let's look at stain index. Stain index tells me resolution. So the higher the value, the better resolution. What we are seeing on the green line is that if I decrease the gains, resolution is expected. But if I increase the gains by 50, that doesn't change much the calculation, because I'm also amplifying the background. Point is that if you look at the shape of those curves for optimal plus 50 minus 50, it's the same. So if I want to define the optimal titer, if we did it through those automated calculations, the titer was the same, around 125 nanograms in this case. Percentage didn't change, expected for CD4, but important to point it out. It's a highly expressed marker. Percentage is not going to be that informative, but useful still to have the information. So if I look at molecules that are not that high expressed, like CD314, I mentioned this before, that co-staining is going to be useful to know where the positives are if they are not that highly expressed. So we did co-staining with CD8. And then these are the concatenated images. Once again, we see an increase in the positives, decrease if I lower the gains, and the optimal titers right in the middle. Let's see the impact of that. What we are seeing, again, lowering the gains will reduce your resolution. We see that green line for the stain index. The optimal titer was the same, independently of the settings. And finally, the positivity, percentage of positivity, it's now playing a role. Because if I go down to 250, 500, you see how your ability to capture all the positives is decreased. So for this one, we would go with the highest. And an observation from this is that this titration curve is never reaching a saturation, and that's something common that we saw. Not as common, but very reflected based on the markers. So my observation for the dimly expressed markers. If it's a dimly expressed marker and you don't reach saturation, then once again, there's going to be a lot of factors that if you're not at saturation will impact the quality of your staining, especially if you want to do quantification. So you might want to have consistency in the protocol, very strict staining protocol in terms of all the variables that I mentioned. One more example of a dimly expressed marker, that if you reduce the gains, then you might not be able to see the population. This is for TCR gamma delta. We co-stain with CD3 in order to find better the positives. And here, the concatenated images for changes in the gains. If we look at the plots, same antibody concentration, very similar observations as for NKG2D. And it's the fact that if I lower the gains, my resolution is decreased, that based on the value for stain index, but the shape is similar. So the optimal titer would be the same. And once again, how positive, plotting the positivity will definitely give you a better idea of where your concentration needs to be to capture all the positives. So another a non-saturating antibody and how careful you need to be with your staining protocol in that particular case. So we have an answer. Settings for optimal titer don't play much of a role, but definitely they do for your resolution. Now the data types. So if you have a spectral unit or an Aurora or Northern Lights system, you're going to have raw files. And then once you calculate and mixing to understand in a multicolor sample what fluors are present and in what proportion, you're going to have now unmixed data. So does it matter if I do the titration calculation in raw or in unmixed data? Let's look at the data. Short answer is no, doesn't matter. Showing you CD14, we look at the concatenated plots, they look similar, raw versus unmixed. And if I plot stain index, it's exactly the same, not showing percentage of positivity because of time. This is CD57. So again, if we look at these two, look the same. If we look at the titration curve, looks very similar. And once we define optimal concentration, was exactly the same value. To show you other examples, CD8, HLA-DR, the same. Doesn't really matter if we use and raw versus unmixed. For HLA-DR, one thing that I wanted to point out is that this one is also not reaching saturation, and we have seen that. The problem with this one is that if you, for the low express antigen, something I forgot to mention, you can increase the concentration. For this one, if you increase the concentration, your signal might be out of scale, plus the fact that HLA-DR is a secondary marker, and it's going to add spread into your tertiary ones. Also important for HLA-DR that is going to be expressing multiple populations, where do you need it? If you need it for B cells, probably going lower, it's going to be not that critical. But if you care about HLA-DR in the T cell activated T cells, then you want to capture that shoulder of positivity. So all of that will also depend on what your cells are, cells of interest are, and the levels of expression of a particular molecule in those cells. No effect if you use raw versus unmixed for the optimal titer. What if you extract autofluorescence? Does it matter in terms of the optimal concentration of antibody? So first, here, this part will depend more on the fluorochromes. How much light the detector that it's giving you the signal for a fluorochrome is getting just from unstained cells or from the autofluorescence. And here I'm showing you CD27. If I show you without extraction and extraction, and for the concatenated images, they look very similar, where the positives are and where the negatives are, and the background for the negatives. So expected, when I calculate stain index, the curves are very similar. The optimal titer in that orange dotted line is the same. Let me also show you another example, CD19. We get very similar visuals, and you will see how different things can be if we go to fluors that are now impacted by autofluorescence. Main message is that if the fluor is not impacted, the extraction of AF is not going to change resolution, and the other question, the optimal titer, not affected by it. So if I now show you the two more specificities, FITI, PE5, A10, fluors that are far from where autofluorescence is going to give you the highest signal, no differences. Those stain index curves are looking exactly the same. Autofluorescence is not giving any extra added information or benefits. Once again, HLA-DR, how you're not getting saturation, but increasing the concentration is going to be detrimental. So making sure that your staining protocol is very strict, it's critical there. What we are now going to be seeing is fluorochromes are impacted by autofluorescence, where we are getting a lot of background just from in unstained cells. This is CD16. It's a good example because cysteine is expressed in multiple subsets, right? Monocytes, lymphocytes, so we'll see the differences there as well. This time, if I show you no extraction versus extraction, and we are seeing, we can tell how in no extraction, the negatives are really high in intensity. So that makes my ability to separate from the positives a little bit more difficult, less resolution, versus the AF extraction, where the negatives are down to 0 because the AF was mathematically extracted. That's reflected in the stain index curves. We are seeing light green on top is when we extracted autofluorescence, higher stain index values, higher resolution expected, clearly seen on the plots. But when we define optimal titer based on the curve, based on the saturation point, that's exactly the same. So AF is not important for the optimal titer, but it is for resolution. Now, if we look at lymphocytes, less autofluorescence, but still high levels on BUV 496. So we see those negatives on the lower left plot. We see those negatives a little bit higher, not right around 0. Once that's extracted, we see on the right, the negatives lower, that translate in the plot into higher stain index, better resolution, but once again, the titer was the same. And important for this particular one is the fact that now my optimal titer for monocytes is different from what I need in lymphocytes because they have different levels of expression of that molecule. And it's going to be the same if you had, for example, whole blood with now the granulocytes being there, expressing CD16 in higher amounts and maybe taking most of the antibodies. So important to take that into account, what populations are expressing and how that's changing others. Other examples in BUV dyes, because that's where most of the AF is coming into those detectors for those dyes, primary detectors for those dyes. We go from no extraction to AF extraction. If we look at the concatenated images, we see the difference visually, and that goes into the calculation. Same conclusion. Extracting autofluorescence improves your resolution, but when you are defining the optimal titer, it's not really playing a role because the titration curves depends on the molecule, on the antigen that you are capturing with those antibodies. So it's going to be the same. Another example, again, tends to be for molecules that are low expressed, CCR7. So an option there would be to go higher, 2 micrograms per test to get better that saturation point. Another question we get is, does it matter if I'm using whole blood or PBMCs? Does it matter if I use intracellular staining or surface staining? So let's take a look at that. The first comparison we did was whole blood versus PBMCs to evaluate markers expressed in the lymphocyte population. Here, it's gonna, like autofluorescence effect will depend on the fluorochrome. Here, the differences will depend on the marker and how any of the steps in the sample preparation could affect the antigen expression, the integrity of those antigens, and finally the signal. For example, for CD45 conjugated to Alexa-Fluor 488, if I show you the same cells titrated with the same antibody with the different concentrations, but obtained just through PBMCs or whole blood, there's no big differences. That CD45 array looks very similar. And that goes into the curve on the right. The stain index is very similar. Same thing with the optimal titer. Another example could be CD3, labeled with a dim dye, Spark Blue 550. What we are seeing is that clearly in PBMCs, the separation is lower, just in the concatenated image. If you go to the whole blood, now the separation is better. So there is an impact on how the cells are prepped before you stain. And we see that on the stain index. Now, 2 observations. In whole blood for CD3, the stain index was definitely higher than PBMCs. And the other observation is because of the shape of those titration curves is different, then the optimal titer will be different. So important to consider. Or just the observation that for your markers, for the titration evaluation, you want to follow the same protocol, use the same cells, the same way they are stained or stimulated or prepped. And finally, the same staining protocol to have a match on whatever could affect the binding, in this case, related to the antigen. To show you CD127, CCR7, once again, just if we look at the stain index curves, we see how different they are versus the pattern we have seen before where things match perfectly. This is not the case. So they are not the same. And then the optimal titer, it's calculated differently. So important to consider once again that anything you do to the cells before titration could affect your patterns and finally your optimal titer. Another thing that we evaluated is, let's say you stain for CD4, and after that you will go on one hand to surface staining only. And the other one, CD4 is a stain, but then you will go through fixation, permeabilization to get access to some intracellular targets of other specificities. Let's see the results. Just from the scatter plot, we can tell that there are differences. So that also, it's going to be impacting other things like markers. Showing you CD4 conjugated to Alexa Fluor 647. Once again, here marker is what's critical. And this is CD4 stain on both for surface, just following different steps after that surface staining. What we are seeing is with surface, CD4, as we were seeing before, is separating well. For intracellular, it's not. The positives are spread, dimmer intensities, less separation. And if we look at the stain graph, stain index graph, we are seeing that like dimmer intensities are translated into a lower stain index, much better in surface and different titers, which then answers that question that stain protocol is going to play a critical role. In that case, it's how whatever it's done during the staining protocol, fixation of perm, it's affecting your molecules of interest and finally the quality of the staining. Another example, HLA-DR conjugated to PE FIRE A10. We're seeing concatenated image on the left, beautiful staining, good separation for your positives. On the right, for the intracellular, no staining of HLA-DR, where the intracellular protocol, that's heavily impact. And the impact on both examples, it's mainly on the positives. If we look at stain index, it's much better. Clearly, we see that on surface. And again, in this case, optimal titer was the same. It's a non-saturated antibody. But how I'm being able to see the dimly expressors for HLA-DR in the surface, but not really clearly on the intracellular. So things to consider, and that's why use a metric that allows you to assess resolution, define percentage of positive, it's a low expressor, and for HLA-DR, and in general, for any marker, make sure you have the concatenated image to guide your decisions. Now showing you the same marker, CD8, how different it could be using the same clone just across conjugates. Surface, on top, BUV805, surface versus intracellular. They look pretty similar when we compare those concatenated plots. In terms of stain index, also similar. Same optimal concentration, a little bit better for the surface staining since the positives are tighter, or the MF5 is going to be higher. The CD8, what we are seeing is when I go from surface for intracellular in the pseudocolor plots, what you're seeing is that the background, it's increasing. So the opposite to where we were seeing in those previous specificities. Now it's the negatives that are increasing. So another potential impact of changes in your staining protocol that it's important to control for. Changes in the optimal titer for CD8, 395, but across similar in general for all of them being the same molecule around 250. What we are getting now, it's to the last comparison. Frequent question. I was using a conventional, do I need to retitrate if I'm using now an Aurora? So let's see the answers. This is an example. This is CD4 PE. You see multiple concentrations tested, the concatenated image for the Aurora system versus a 4-laser conventional cytometer. They look similar in the shape, right? The positives are a little bit dimmer on the Aurora system, brighter on the other one. The negatives are tighter on the Aurora and more spread in the conventional. Just to in something I want to point out, for this we use cytic assay settings in the Aurora system and optimized settings for the conventional, making sure that the negative population was away from the electronic noise in the system. The point is that since there are differences in the intensity of the positive and in the spread of the negative, that compensates, and what we get at the end is that stain index looks very similar. For this particular conjugate, we see the curve. similar curve, similar values. So in this case, for both instruments, the concentration, similar to the observations we had before for the specificity, is that we are getting optimal concentration at 32 nanograms per test, independently of the instrument. So if I now go to a low express marker, because if an optical configuration or an optical layout has better efficiency or better sensitivity, then I will be able to see better dimly expressed markers. So if you look at on the left, the Aurora system, titration for TCR gamma delta, what we are seeing once again is that 16 nanograms. That's difficult to tell, still doable, still separating. And then we go and increase the concentration and see that increase in MFI, reaching a point where you get the highest, but not clearly seeing the saturation. And lower in the negatives, not highly impacted. It's far from the issues in terms of PE fire 700. On the other plot, we are seeing a 4-laser conventional. In terms of intensity, very similar, a little bit more spread. And because of that, when we calculate stain index, what we are seeing is the dark green, it's the spectral cytometer. Values for that stain index are higher. Lower resolution for the conventional. But at the end, if the question was, is the optimal titer changing across instrument? It's not the case. And it would make sense. The optimal titer depends on the binding between an antigen and antibody and how much antibody you need to capture the expression of that marker. So supporting that, there's no changes. Important, again, also when you have a low express marker or a low frequency population, that you monitor percentage of positives. In this case, again, if you go lower, you will not be seeing all the positivity in your sample, which is something critical for every panel. Now showing you NKG2D. Aurora system versus a 4-laser conventional, no big changes, slightly lower on the spectral in terms of stain index, but the percentage of positive, once again, what's going to guide with an antibody that is not reaching saturation, because if you go way too lower, you're losing a lot of those gated cells, and this is gating out of the CD8 positive population as a co-stain. Few more examples. This is done in mouse samples, and now comparing an Aurora system and 2 conventional ones, different configurations. And this is for CD4 APC Fire 750. So what we are seeing is the full picture of those titrations done on different instruments. We see changes in intensity, changes in separation, changes in the background for the negatives. But let's look at the data and the stain index. For this particular one, expected because of the detectors that we use and the low gains and also low noise, what we are observing is that the Aurora system gives you higher resolution in terms of the number for that stain index value. But the concentration that is needed to separate better that positive population with low background, high MFI is the same across instrument, or was the same across instrument. And this is a constant observation that we saw in other data sets. Here, just showing you some representative data. And one more, this time for APC, comparing multiple instruments, spectral versus conventional. Again, the curves look very similar, slightly a better resolution for the spectral system with this particular conjugate, but once again, similar definition of what's your optimal amount of antibody added to your samples. So that's the end of this presentation. Some take-home messages, starting with the fact, I hope this proves you this point well, that titration is so fundamental for successful experiments in flow cytometry, and that you need to plan carefully every step of the titration process to have good results at the end. In terms of the conclusions from the testing, is that optimal antibody titer selection will not be impacted, or was not impacted by the metric that we use to calculate resolution, by the instrument setup going higher or lower, by the data file type, raw versus unmixed, by extracting autofluorescence, or by what instrument you're using or which technology. Important to emphasize something that we already knew, but part of the conclusions, that the use of full spectrum profiling, or the Aurora, Northern Lights systems, Cytec assay settings, the autofluorescence extraction has definitely an impact on having better resolution of your data, easier separation. And finally, that the optimal antibody concentration depends highly on your cells. So anything that goes in your cells in terms of cell preparation, staining protocol, you want to assess for every marker separately and define what the better conditions are to get good resolution as well. Well, thank you so much for your attention. I'll be happy to take any questions. Also, thank you so much for an amazing internal team in the applications group that helped generate this files and all these data sets. And also thank you to FCS Express, because they save us a lot of time and simplify and make our workflow more efficient by using that titration template. Well, thank you again, and happy to answer any questions.
Thank you, Deanna, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have any questions you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have the time for. So let's get started. Our first question is, what do we do if the titration never shows saturation?
Yeah, so that few of the examples that we had show that, right? And it can be the fact that the manufacturer is not really giving you a concentration that it's at a saturation point. So you could go higher. So if the recommended or volume is, let's say, 5 microliters, you could test 10 microliters, or check on the concentration and go higher, 5 micrograms, usually around 2 and 1 and 2 micrograms per test. It should be enough for most of the antibodies. But yeah, if you don't get to that point, options that your manufacturer gives you a more concentrated antibody, something we are working on. Number 2, understanding that if it's not at saturation, the contribution of other variables, it's going to be higher. So having a very strict protocol in terms of your staining, controlling all the variables that could play a role. Once again, temperature, titer, the number of cells that you're using, more than titer, the incubation times, all of those things that could add to the variation in the data, and as well as technical errors, right? Pipetting, so making sure your pipettes are calibrated, things like that could help with. The other thing that could, if it's a highly expressed market like HLA-DR, you don't reach saturation. If you go higher, as I was recommended, things will get worse. You will get more spread and your signal will be off scale. So in that case, having a competitive assay where you add unconjugated antibody in order to reach that saturation in a different way, adding more antibody that is not having the fluorochrome in there to get to that plateau, that would be another option.
Wonderful. And our next question is, do you have any recommendations on how to titrate viability dyes?
That's a point that we didn't talk about here. And part of what I was saying for titration of antibodies is that you want your positives really high, saturation point, but your negatives low. How low? Ideally at the same level that when you run unstained cells. So that would apply for viability dyes as well. The optimal titer in my hands would be where your negatives are not giving you that high background, really close to unstained, but definitely something that you would need to test. In my experience, most of the viability dyes, the suggested concentration is way too high. So also depending on the sensitivity of your instrument, you might need to go lower, but I would do it making sure you have enough dead cells and that you have a good negative control for that. Positives and negatives, it's going to be critical. And also, the fact that you want your negatives really low, it's going to be important, especially for amine reactive viability dyes.
Wonderful. And it looks like we have time for one more question. What would you suggest if I would like to titrate a marker that is homogeneously expressed on all my cells, so I'm missing a negative population?
Yeah, that's a good point. Can be the problem with CD45. So you want your negatives to be in contact with the antibody so you can assess part of what the negative population will be about. So I was saying the negatives the signal you get from the negatives will depend on your instrument, the noise, or the instrument settings, how high your gains are, how optimized they are. It will depend on your cells, the autofluorescence, and it's gonna also depend on your fluorochrome, the non-specific binding. So making sure you control for those. Gosh, I got lost. Can you repeat the question? I was saying too many things.
No
Thank
worries. The question was, what would you suggest if I would like to titrate a marker that is homogeneously expressed on all my cells?
Thank you. So in that case, one suggestion would be use unstained cells, spike some unstained cells. Issue with that is that you will still have autofluorescence idea, the instrument background idea, but you will not be able to assess for the non-specific binding of that antibody to anything that is present in your cell that is not the specific target of interest. So that would be the caveat with using spiking unstained cells. But I guess that would be the only solution, or find a tissue or a sample type that will allow you to test that better. Once again, ideally, you want to do the titration in the same cells that you are planning to use in a multicolor experiment. Thank you.
Thank you, Deanna. Do you have any final comments for our audience?
No, that if we can provide all this information, we are going to be providing a white paper with all this to our customers soon. So stay tuned. Thank you so much for staying and your participation. And thank you to the organizers as well.
Thank you again, Deanna, for your time today and your important research. We would also like to thank LabRoots and our sponsor, SciTech Biosciences, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of the registration. This webcast can also be viewed on demand. LabRoots will alert you via email when it is available for replay. We encourage you to share that email with your colleagues who have missed today's event. Until next time, bye-bye.